Have you ever pondered the potential of gene editing? Could we alter the building blocks of life to eliminate disease or enhance our abilities? Welcome to the fascinating frontier of genetic engineering where science fiction meets reality. Imagine a world where we can rewrite the genetic code, just like editing a sentence in a word processor. This is not a far-off dream, but a rapidly emerging reality. With the advent of revolutionary tools like CRISPR, we are on the brink of redefining what it means to edit our genetic makeup. The potential is enormous. Picture a world free from genetic disorders where diseases like cystic fibrosis and Huntington's disease are things of the past. Or consider the tantalizing prospect of enhancing human abilities, making us faster, stronger, or even smarter. Gene editing seems like a concept from a science fiction novel, but it is real and happening right now. But how does it work? Imagine a computer program where you can easily cut, copy, and paste text. Now envision doing the same with our DNA. Let's break down the complex process of gene editing into simple, understandable steps. First, consider the double helix structure of our DNA, the blueprint of our existence. It's like a long, twisted ladder, with the rungs of the ladder made up of pairs of 4 different molecules, or bases. These bases, known as adenine, cytosine, guanine, and thymine, are the letters that spell out our genetic code. Now imagine a tool that can precisely target a specific sequence of these letters, much like using the find function in a text editor to locate a specific word or phrase. This is the role of a molecule called CRISPR-Cas9, which is often referred to as molecular scissors. CRISPR-Cas9 is a naturally occurring defense mechanism in bacteria, which scientists have repurposed for gene editing. It works by guiding the Cas9 protein to a specific location in the DNA sequence. The Cas9 then acts like a pair of scissors, cutting the DNA strand at that exact location. This break in the DNA prompts the cell's natural repair mechanism to kick in. Here's where the copy and paste analogy comes into play. Scientists can introduce a piece of DNA that carries the desired genetic information into the cell. As the cell repairs the break, it can incorporate this new piece of DNA into its own sequence. This allows scientists to insert new genes or modify existing ones at this location. Alternatively, if the scientists don't provide a new piece of DNA, the cell will try to repair the break on its own, often making mistakes in the process. This can disrupt the function of a specific gene, which can be useful if that gene is causing problems. So, just like a text editor on a computer, gene editing tools allow scientists to modify the text of our DNA. Harnessing the power of gene editing could potentially eradicate genetic disorders and enhance human traits. But what are the implications? Imagine a world where we can cure genetic diseases like Huntington's, cystic fibrosis, or even some forms of cancer. The promises of gene editing are truly revolutionary. It could allow us to enhance human capabilities. Imagine if we could make ourselves more resistant to disease, or even slow down the aging process. But with great power comes great responsibility. There are serious ethical and safety considerations to think about. For instance, who gets to decide which traits are desirable? And what happens if gene editing technology falls into the wrong hands? Could we end up with a society divided between those who can afford to upgrade their genes and those who can't? Furthermore, the technology is still in its infancy. We're not yet sure of the long-term effects of editing our genes. Gene editing holds immense potential, but like all powerful tools, it must be used responsibly. So, what have we learned about gene editing? Let's recap. We've delved into the frontier of genetic engineering, where gene editing stands as a groundbreaking technique. This process, akin to a computer's copy-paste function, involves modifying DNA sequences to alter genetic traits. It's like rewriting the very code of life. We've also simplified the process of gene editing, breaking it down to its core. With tools like CRISPR, scientists can selectively edit genes, offering hope for the treatment of genetic diseases and conditions. But like any great innovation, it doesn't come without challenges. We discussed how ethical concerns, technical limitations, and potential unforeseen consequences stand as significant hurdles in the path of this revolutionary technology. The future of gene editing is still being written, but one thing is clear: it has the potential to shape the future of humanity in ways we can only begin to imagine.